It's a pleasure to be here. I'm not really a methodologist, but sometimes I dabble in methods, and sometimes the dabbling leads to long-term research projects, and this is sort of an example of that. And this is really, the work that I'm going to talk about today was really my attempt to grapple with what I think is a kind of fundamental issue with how we approach brain imaging analysis. And actually this work, I kind of tapered off this, but really Jeremy was the one who picked up the torch and ran with it, and so he's been doing a lot more with it recently than I have. And so really, if you get interested in this, you should talk to him rather than me. And I should say also that this is a little bit backwards in the sense that I'm going to start with talking about some fairly heavy machine learning concepts. But then in the morning, in the later tutorial session, I'm going to do a much gentler introduction to basic modeling issues. So if some of the things that I talk about here seem very mystifying or confusing, we'll have time to go through them more leisurely later. And of course, I'm happy to field any questions. I'm going to try to keep this short so that we have plenty of time for questions. Okay, so pretty much everyone, I'm sure, is familiar with the general pipeline for fMRI analysis. And what I want is to call attention to a particular aspect of this pipeline. That's the idea that we measure fMRI in the unit of voxels, right? So we've broken down the 3D image into these little cubes. And then what we're going to do is take so that's our measurement unit, right? And then we've defined an analytical unit where we fit statistical models to the data, where the parameters of that statistical model are tied also to voxels, right? So we have voxel-specific parameter estimates in our statistical models. So we have this connection here between our measurement unit and our analytical unit, right? That they're basically the same thing in the standard pipeline of analysis. But that actually doesn't have to be the case. In fact, there's good reason to argue that that shouldn't be the case, right? So what I want to, the main message I want to convey to you is that you should resist the tyranny of the voxel. The unit of measurement need not be the unit of analysis. Because of course, there's a lot of stuff going on inside that voxel. And the stuff that's going on inside the voxel and beyond a single voxel are really the things that we care about. And there's nothing really special about voxels. That's just how we happen to measure the data. So really, we want to design analysis methods that get away from thinking in voxel-based terms. So here are a few of the issues that arise. So we've imposed this discretization of the anatomical space, and we've now elided this measurement discretization into a parametric discretization. What that means is that if I change the discretization of the anatomical space, like say doubling the number of voxels, now I've doubled the number of parameters that I have to estimate, right? That seems really wrong, right? Why should the measurement discretization impose these constraints on the parametric discretization? And of course, that leads to things like there's an explosion of parameters to estimate. There's issues of that it's harder to align results across subjects. And I think most importantly for me, the parameters should really reflect meaningful theoretical quantities, right? So when we write down a statistical model of anything, what we really want to be going towards as scientists is some kind of hypothesis about the generative process, right? What was the generative process that gave rise to the data? And we as scientists are collecting experimental data and trying to invert that generative process to recover the underlying theoretical quantities that we're interested in. We're not just doing kind of black box data analysis because that's less likely to lead to scientific insights. So what I'm going to talk about is a new approach. Well, I guess it's not really that new anymore. I did this a few years ago. And the basic idea is that the observed neural data arises from a superposition of basis images. Now that by itself is not that new. People have been doing this for many years. But the key idea here is that these basis images are going to be topographic and voxel free. So the way that we're going to define the basis images, the basis functions is completely independent of the measurement discretization. And I'll explain more what I mean by that in a moment. And we're going to also, much like in the standard analysis pipeline, assume that the superposition is covariate dependent. So which of these basis images is active depends on the current covariance. So let me first talk about the basis images. Really, I should be calling these basis functions because the images, right, are in the discretized space. However, we've chosen the discretization into voxels, but the basis functions live in some continuous anatomical space that is independent of whatever discretization you impose on the data. So what I'm going to do is define a function where k here indexes the basis functions and v indexes voxels. And this function is going to map an underlying topographic latent source into a pattern that you can observe in the measurement space. And in fact, I don't place any constraints on the particular functional form, but this is the one that we've used most frequently, and this is just a radial basis function. So the idea is that each source, each latent source is parametrized by some mean that's defined in coordinates in the anatomical space, and then some width of that basis function. So in the simplest case, we assume it's symmetric. So it's basically this sphere-like blob that's positioned at some location in the anatomical space, and we have a bunch of these different blobs with different parameters, so different widths and different locations, and we're going to model the observed neural data as a covariate dependent superposition of these blobs. And the inference problem will then be to recover both the weights that determine the superposition and the underlying parameters, so the mu's and lambdas. Okay. Here, just a notational point. I'm not going to show too much math here, so just kind of follow along the basic concepts. The R here is the three-dimensional coordinate of that voxel. And one thing that I'll show you later is that basically the exact same approach can be applied with very little modification to spatial temporal data like EEG and MEG. Okay. So we're going to call this topographic latent source analysis. Because we're modeling these latent sources as these topographic entities. All right, so here's the generative process in a nutshell. So we have this neural data. We've collected n images, each of which is v voxels. And we have, just like in standard fMRI analysis, we've constructed some design matrix where we have C covariates. And then we're going to imagine that we have k of these underlying latent sources. And so this F matrix is constructed by applying each latent source and mapping each latent source into the voxel space. And then taking some linear combination of those latent source images, those mapped images, according to this W matrix. So here's the schematic. You can imagine that there are these little blobs here. Those are the different latent sources. And then we're taking some weighted combination to generate the prediction of the observed neural data, right? And so that gives us something to compute a likelihood function on. So we can ask, to what extent does this superposition look like the actual neural data? And we can try to then use that likelihood function to invert the generative model and recover the underlying parameters, the W matrix, and the parameters of these basis images. So notice that in this framework, there are no parameters that are specific to voxels. It's completely voxel independent. So if you double the number of voxels, you have the same number of parameters. I think that's very important. However, you can recover the kind of traditional fMRI mass univariate analysis if you think about basis images, basis functions that are whose width basically shrinks down to a single voxel. So now you have, now basically each latent source corresponds to a single voxel, and then you take k to be the same number as v. So we have basically, if I just construct basis images that are restricted to a single voxel and I exhaustively cover all the voxels, then what I've done now this basically becomes an identity matrix, and I've recovered the standard mass univariate GLM. Okay. There's another issue here that arises. So in standard fMRI analysis, we tie ourselves into knots trying to align subjects into the same anatomical space, and that at the end of the day corresponds to aligning voxels across subjects. In TLSA, it's a little bit different, right? Because now I've defined these sources, and there's nothing in the model that I've described to you so far that enforces that different subjects have the same latent sources. So the way to deal with this issue is to define a hierarchical model. So what we're going to say is that each subject level source, I'm just showing this in 2D for simplicity, each subject source is drawn from some group level distribution. So they're going to be centered around this distribution, but there can be some anatomical variability in where that subject's latent source is centered. And then what we're going to do is to try to recover both the group level distribution and the individual subject level sources. So how do we actually do this? This is kind of a thorny question. And actually, this was, writing down the generative model is easy. Inverting it is actually quite difficult and complicated, and I won't bore you with the details. Basically, what we want to do is recover, as I mentioned already, we have some covariates and some neural data, and we want to recover theta, the parameters. Remember that theta corresponds to that weight matrix and then the parameters of the latent source. And so Bayes' rule tells us that that's going to be the product of a likelihood and a prior. Now the problem is that this is completely intractable, horrendously intractable. However, we can do a machine learning trick to simplify this process, or well, it's simple to use, it's not simple to derive. And the basic idea is that we're going to approximate this posterior distribution with a distribution Q, and Q has its own set of parameters which we're going to try to optimize. So essentially what we're going to do is map this inference problem into an optimization problem. And the optimization problem looks like this. So we're going to try to choose the approximate distribution Q that minimizes the Kobach-Leibler divergence between the approximation and the true posterior distribution. And so that minimizing Q is going to be denoted Q star. Now, if you're completely unconstrained in this minimization, then you'll just recover the exact posterior, right? But that's not very helpful, right? Because we already said that optimization is intractable. However, we can impose some constraints. So in particular, we can assume that this approximate posterior factorizes in a particular way, and we make some other distributional constraints. And when you do that, then you can basically derive very efficient updates for the parameters of this approximate posterior. And I spent a long time reading really boring linear algebra texts to figure out how to do this efficiently, to the point that I could run all these things on my laptop, and it's almost as efficient as just fitting a GLM. Okay, and this is just for the connoisseurs out there, this is known as variational Bayes. This is a variational optimization problem. So here's an example of what it looks like. So I've just taken two conditions from this MRI experiment. This is just the raw difference between the means of the two conditions. This is what it looks like when you take the TLSA images and contrast them. So it gives you this, and then you threshold them based on posterior probabilities. So you get these very localized clusters. This is what you do when you run a spatially regularized GLM. So this is kind of like the normal GLM, but in SPM you can do this fancier variational approximation of this smooth GLM. And what you get is something that looks kind of similar, but you can see that there's a lot of mess around here too. Now, whether or not that is good mess or bad mess is an empirical question, right? Just looking at these maps doesn't tell us. What we really need to do is some quantitative assessment of how good this is, and some agreed upon metrics. So I focused on two metrics. One is reconstruction. So given a set of covariance, how well can we predict the neural data? And then decoding, given the neural data, how well can we predict the covariance? And all the analysis I'm going to tell you about are based on cross-validation. Sorry, this is a little bit small. Let me just summarize what this is showing to you. So this is showing classification accuracy. And basically, TLSA and the hierarchical version of TLSA that's shown in the purple line get more or less about the same accuracy as using a discriminative algorithm like regularized logistic regression, and do quite a bit better than alternative generative models like Gaussian naive Bayes, which is basically assuming some factorization of the box. So if all the boxes are independent, then you get back Gaussian naive Bayes. Reconstruction, oh, and I'll mention one other thing about this, which is that we don't really see much of an advantage of the hierarchical version of TLSA compared to the non-hierarchical version, which was kind of disappointing to me because I was a big believer in hierarchical models, and actually at the end of the day it doesn't really make a difference. The one place, as I mentioned, that it does make a difference is, of course, interpretability, because if you're modeling multi-subject data, you cannot interpret the sources unless you use this hierarchical model where that imposes some consistency across subjects and their sources. So here's reconstruction performance. So again, this is, I've gotten the covariates and I want to reconstruct the neural data. And again, you basically do a lot better than this Gaussian naive Bayes. In this case, we did some dimensionality reduction for naive Bayes. And again, the hierarchical and non-hierarchical versions do about the same. So I want to talk now about a few different extensions of the basic modeling framework. So the first is to spatial temporal data like EEG and MEG. And the idea is really to take exactly the same concept that I introduced to you already, but now define the basis functions over both space and time. So if we have EEG data that you can model it either 2D or 3D, and then you add a fourth temporal dimension, now we can get a model that looks something like this. So I've defined the spatial temporal basis function, and the exact model works the same way. So I think for the applied session later today, I think we're going to play around with the toolbox that I wrote for this, and the exact same algorithms apply the exact same functions apply for both EEG and fMRI data. The only thing that changes is how you define the anatomical or spatial temporal positions of the different measurement units. So you have to tell it that here there's this electrode at time t, or in the fMRI case, here's this voxel in this three-dimensional coordinate space, and then the exact same algorithms apply. Yeah?
Just a clarification. Why was the naive phase getting worse with increased number of components? Just intuitively.
Probably because, so the number of components there corresponds to the number of singular values or principal components that are retained in the naive base. So essentially it's overfitting as you get more components. Oh yeah, and that reminds me, which is that another thing you might have noticed here is that the performance of TLSA doesn't depend strongly on the number of components. Basically, once you get some appreciable number of components, performance kind of flattens out. So that's reassuring that there's some robustness, I think, to the particular parametric choices of the model. So we might ask, can we use this technique to recover some well-known event-related potentials using EEG? So here's an oddball data set that was collected by Per Sederberg. And so the key contrast is looking at rare trials versus common trials. So in these oddball tasks, you occasionally will get some stimulus presented that is uncommon. That's the rare stimulus. And then you get this this P300 effect, which we can see right over here, for the rare minus common. And so basically the TLSA recovers this, but oh, what just happened? So TLSA recovers basically a smooth version of this, which is I think quite nice. And again, we can apply classification and reconstruction metric to this. And just to make a long story short, you do get better performance of this algorithm compared to naive Bayes. And there's not much difference between hierarchical and non-hierarchical. And there is not much difference as a function of the number of components that you use, the number of sources. And we can also compare with other, you might say, well, naive Bayes, who uses naive Bayes? Let's compare against some actual, state-of-the-art discriminative algorithms. So one is regularized logistic regression. Another is this bilinear components analysis that was developed by Mads Deerholm, specifically for analyzing EEG data. And here you again see that you get some performance advantage for TLSA. And I've also applied this to some brain-computer interface data sets. This is the motor imagery data set. And it doesn't do quite as well as the state-of-the-art, but it basically is statistically indistinguishable from the state-of-the-art algorithm. So I want to talk, the last part of my talk, I want to talk about some non-parametric extensions of this. So the idea here is that we've made some fairly constraining assumptions about the structure of the model. We've constrained the parametric form of the latent sources, and we've constrained the number of latent sources. You have to specify both of those things when you run the algorithm. Can we relax those constraints? And in here, I'm going to appeal to Bayesian and non-parametric modeling ideas from the statistics literature. So for modeling more flexible sources, I'm going to appeal to this concept of a Gaussian process, and to model unknown number of sources, I'm going to appeal to this concept of a beta process, and I'll explain at least at a superficial level what those mean. Okay, so how do we construct more flexible latent source functions? What we're going to do is place a prior over those surfaces that allows a wide range of shapes but prefers a smooth one. And that's basically what a Gaussian process is useful for. The idea here is that a draw from a Gaussian process is a function. So a Gaussian process is much like a Gaussian distribution is defined by a mean vector and a covariance matrix. We can define a Gaussian process as parameterized by a mean function and a covariance function. So it's like applying, it's like defining a Gaussian distribution when there's an infinite number of dimensions. And if that's too abstract for you, the key point here is that the covariance function defines the smoothness of the underlying function, and the mean function defines its shape. So what we can basically do is define the mean function in terms of these parametric sources. So like we say that they have to be roughly spherical, but then the covariance function allows some deviation from that mean function, and such that it looks kind of like a sphere, but it will allow some more irregular shapes around it. And what we can do then is we can optimize the parameters of the Gaussian process, so we can basically infer what's the right level of smoothness to impose on the data. This is just a technical point, which I'll skip. And I've applied this to retina topic orientation maps in V1 that were collected by Jeremy Freeman. So I'll show you the data in a moment. The key, I just want to give you an intuition for what this looks like. So here's the standard radial basis function. And here's where you have a Gaussian process source. And what you can see is that it still looks a lot like the radial basis function, but it's got this irregular stuff here that is smoothly varying over space. And that kind of irregular, smooth stuff is important for capturing things like retinotopy data, where it doesn't look, the data don't look particularly radial. So we can capture these kinds of irregularly shaped but smooth surfaces using using this Gaussian process prior over latent sources. And so what I'm showing you under here is the underlying reconstruction of these discretized data points. And we can look at this more quantitatively and ask how well can we reconstruct the orientation maps in a cross-validated manner from this model, and we can compare it to the standard map estimation of TLSA with the parametric sources, and compare that to this Gaussian process TLSA, which does better than both the traditional representation and naive betas. And sort of a similar story with classification performances, where we're trying to decode the underlying orientations. All right, second problem, how do we have more flexibility in terms of number of sources? So I told you we had to specify beforehand how many sources we want. Nonparametric models give us a way around this problem in a kind of counterintuitive way. So what I'm basically going to tell you is, imagine that we have an infinite number of sources, but now I'm going to impose a sparse prior on those sources. So even though I have technically an infinite number of these sources, my prior says that only a small number of them are going to actually be active in my data set. And so the sparseness prior basically dictates how many sources I have in expectation. So the way to construct this is what's sometimes known as a spike and slab model. So instead of the Gaussian prior on the weight matrix that we were assuming before, I'm going to define the spike and slab prior, which is a mixture of a Gaussian distribution and a delta function at 0. So what the delta function at 0 says is that this source is basically off. It doesn't count. Even though in some technical sense it exists, it's not being used in our data. So the more of the sources that have this delta function, the sparser and the fewer sources that you're going to have. And we basically model this as this product. We basically decompose the weight into the product of this spike z and the slab, normally distributed slab u. And the idea now is to do inference over this decomposed weight variable to figure out how many of the sources have some non-zero component, spike component, right? How many sources are on? And this is just illustrating this on simulated data. So I can generate data with a known number of sources. In this case, it's 5 sources, and you can show that I didn't mention that the technical term for this is called a beta process, and what you can see is that the model effectively recovers the true underlying number of sources. Sorry, I just noticed that the thing, yeah. Okay, okay, so to wrap up, TLSA was designed to capture some important ideas about statistical structure of fMRI data, the most important being that we should decouple the measurement discretization from the parametric discretization and define something that we think more closely approximates the underlying generative process, that there's actually these latent sources with some point spread function that are generating the patterns in the neural data, and then we're going to try to recover those using Bayesian inference, and we can apply this setup to all sorts of different tasks like classification, regression, reconstruction, even hypothesis testing. So you can do traditional hypothesis testing within this framework. And I showed you how you can extend this to EEG to model spatial temporal sources, and several non-parametric extensions to deal with more flexible latent sources and an unknown number of latent sources. So if you remember one thing from this talk, it's to resist the tyranny of the voxel, and don't. It's not all about the voxel. We have to get away from the voxel and get closer towards what we think are more plausible generative hypotheses about fMRI data. And with that, I'll just thank my collaborators on this. At the time, we were basically all at Princeton, but since then, various people have moved to different places. And I'd like to thank you for listening. Yes, yes. Yes, Chris.
If we had data where we had, let's say, 20 subjects but non-overlapping subsets of the brain sampled with each subject.
Yeah.
Would the hierarchical version of the model also allow us to kind of flexibly combine the data across participants without having to directly correlate them to one another?
Yeah, so the question is, if we had a bunch of different subjects that have different subsets of the brain sampled, I'm assuming this is because it's ECOG data, then can this method be used to basically combine or fuse the data without having to try to align all the data, because clearly that's impossible if they're non-overlapping subsets of the brain. And my answer is yes, although I've never tried it, so I guess it's a yes in principle. You'd want to make sure that you don't get somehow weird, spurious sources. But the nice thing about the Bayesian framework is that you can quantify your uncertainty, right? So in a particular subject, if it shows that there's some latent source somewhere in the brain that you didn't actually collect data, and you should, of course, regard that with caution. But yeah, my answer is that in principle, it should be able to combine data. Yeah.
It feels like it's best fitted for univariate effects. So we could replace blobology with spherology,
Yeah.
right? But in neural space.
Yeah.
But what about multivariate patterns,
Yeah.
which will be more finely structured?
Yeah.
Right?
I'm glad you brought that up because this is something that I wanted to mention and forgot. So the question is, we're basically replacing blobology with spherology. So instead of trying to fit univariate models and then recover some blobs from that, some clusters, we're just going to fit the blobs directly in a parametric fashion. And that works fine if you think that the brain is organized in terms of blobs, but it doesn't work so well if you think that there's some fine-grained multivariate structure within a region, right? Because clearly if I define them in this topographic fashion, I'm actively operating against the idea that there's some fine-grained information. So I have two responses to that. One is that oftentimes people use multivariate techniques and it's not obvious that there's actually fine-grained information in the region, right? So for example, we can take brain areas in ventral visual cortex that that seem to be selective for different kinds of object categories, and you can train a classifier on those. And it is true that there may be finely interdigitated information there, but you can also do pretty well detecting a face or not by just taking the global amplitude of FFA activity, for example, right? So in some cases we use multivariate techniques when maybe a blob would suffice. Now obviously that's not universally true, and there's lots of situations where it's demonstrably untrue. So in those situations you really don't want to use a technique like that. If you have any inkling that there's some fine-grained multivariate structure, you don't want to use a technique like this. But I think what's nice about this setup is that you should be able to compare these hypotheses in a principled manner, right? So you should be able to, what I'd ideally like is for everyone to think generatively and to say, I'm going to write down different probabilistic models and encapsulate my generative assumptions for the data, not just run a discriminative classifier, but actually make a hypothesis about how the data were generated, and then compare those things in ways that we know how to do model comparison. I think that's the way forward. Yeah, go ahead.
But in principle, it could be extended, right? So you could model with some assumptions that we have a pattern which gets somehow transformed.
Yeah.
And just model it as well. It's just you would need much more data.
Yeah.
Right? But in principle, it's
so I could write down non-topographic, finely interdigitated link sources. That's certainly within the scope of this framework. Yeah.
I was interested in the sphere part of the spherology. It seems like the radial functions were used for tractability.
Yeah.
But I don't know if you think those are the actual the shape of the latent sources. And so I was really interested in those parameters in that radial function. And I thought we might turn to something like affine transformations on those radial functions, but then we got into these Gaussian process models, but they still look like spheres. It's just a Gaussian process model. Were you surprised by that? Are you interested in allowing these to be more flexible and actually looking at what the latent sources are?
Yeah, so the question is how much do we want to use these kinds of highly constrained latent source parameterizations in terms of the spheres? And even, as you point out, even for the Gaussian process version of this, they still look a lot like spheres. And that's actually by design because I've specified the mean function to be a sphere. So what it's essentially doing is like sphere plus spatially smooth noise. But that might not be a strong enough hypothesis, right? So you're suggesting affine transformation. So basically, can we take a sphere and then warp it in a particular way so it better captures the shape of the data? And absolutely you can. I feel like my memory is fuzzy, but I vaguely feel like I tried to do that at one point. And there's a sort of a graveyard of seemingly good ideas here that I've stopped pursuing.
Cluster time out or
something.
something.
Yeah, exactly. But yeah, I think in principle that's totally reasonable. Yeah.
So say, does your model make any assumptions about what the density of the latent sources has to be constant across the brain? So for example, like the brainstem might be different from like cortex.
Yeah, so the question is, are there constraints on density? The answer is no, but you could write down priors that impose that. Again, that is something that I vaguely feel like I tried to explore, like basically priors for repulsion of sources so they don't get too close and overlapping. I think when that does happen, it's because there's something that's basically non-spherical that the brain is trying to capture by smushing together a bunch of different latent sources. Yeah, I know.
This is a follow-up, I guess, on the multivariate question. So in principle, it's possible to find different latent sources within the same region of interest.
Yeah.
So even if it's a Gaussian process, it could be that a bunch of them, however not adjacent to each other, are generating different sort of patterns. Is that something that you pursue? Because like that would be useful in terms of not just getting some decoding readout, but finding out the underlying structure of different neuronal population behaving in a particular region of interest. Say different orientations in V1.
Sorry, let me just make sure that I've understood your question. So you're asking, is it ever useful that you have basically a bunch of different sources within the same region? Are you asking, is that a good thing that you can draw a scientific inference?
No, given that hierarchical sort of mapping, you should be able to collect different latent sources that are interleaved in space next to each other.
Oh, interleaved? Can you collect sources that are interleaved? Yeah, I mean, in some sense, the source, there's no constraint. The sources can lie on top of each other. Interleaving, I'm not sure exactly how that works, because like the interleaving has to be imposed by the superposition, basically.
So in the neurons, it could be that some populations are right next to each other. Some of them are responding to this, some to this, some to that. But spatially, they're not in different locations. They're all intermixed.
Right.
So what would the process look like for this versus that? Would it be on top of all of them, one, and then another in the same location?
I mean, it would depend a lot on how you parameterize the sources, right? If you need things that are finely interdigitated, you cannot use these kinds of strongly topographic sources. You need to, and maybe there's some topography plus interdigitation. And I guess my answer is that I don't know, and really this is where it gets interesting, right? Because now I've basically written down a modeling framework in which we can parameterize these sources however we want, as long as they're differentiable. So in fact, the software that I've written can take arbitrary source functions. All you need to be able to do is provide it with the matrix of derivatives.
But in this case, it might not be differentiable,
yeah, well, you'll be out of luck if it's not differentiable. But you could. I think that there's no reason you couldn't write down a differentiable model of that sort.
So if, like you were saying, we're trying to move towards like a full generative model description, of trying to generate, the whole activity pattern of the brain and its sources in its full sort of spatio-temporal richness.
Yeah.
So in this formulation, it seems like you could have two sources that are distant but correlated in time, right?
Mm-hmm.
And so this model would, I imagine, still assign different sources because you're constraining them to be spatially localized.
Yeah.
But then you would say, well, that's the job of some next sort of stage of that full generative model to specify the correlation structure, or could that be part of some integrated description at this level already?
I think that's a great idea. Wait, Jeremy, didn't you do something with this?
Yeah. I'll talk
Okay.
about that.
Okay. Yeah. So maybe Jeremy will answer this question. Yeah.
Good idea.
Thanks for the same.
What about, so when you guys fit this to image data, how do you deal with smoothing?
Well, you could. It depends what you're trying to do. I mean, I don't know if I have an absolute answer to that.
I was curious. I mean, so
you can apply to smooth data, or you can apply to unsmooth data. Because the sources are topographic, it shouldn't make too much of a difference, right? Because
that's what I was curious about. Because like in practice, at least when I do it with more decoding methods, the smoothness can make like a huge difference both ways. So like, for things that might be more lower spatial frequency, smoothness helps a lot. But then if it's a higher spatial frequency, then smoothness actually really hurts. And in univariate stuff, if you get a really big SNR boost, if you do the smooth like some sort of smoothing. I'm curious, like if it makes it easier, if it biases you to find sources that are more when you have the smoothing.
Yeah, that's an interesting question. I haven't looked at that systematically, but I mean, I think this is kind of a variant of the same question that has been asked, which is that if you think that there's fine-grained information, then smoothing is going to hurt you, and that's true whether you're doing it in a univariate framework where you're smoothing the data or in a topographic lane source. So again, it just comes down to different hypotheses about how the data were generated. If you can write down another model that doesn't have that smoothness assumption or a weaker smoothness assumption or a different kind of smoothness assumption, you should be able to take this framework and compare those models.
Any other questions for Sam? Crystal clear.
Sorry if that was sorry if that was a little bit intense. The tutorial session will be a lot gentler. Don't give up on me yet.